Aires Üniversitesi Ziraat Fakültesi Hayvansal Üretim Bölümünden Daniel Felipe Salamone bugün bizim konuğumuz. Bölümümüz ile Buenos Aires Üniversitesi Ziraat Fakültesi Hayvansal Üretim Bölümü
İsteyen o daha iyi sesiniz duyuluyor, o yüzden size engel oluyor. Hocam. Yok oradan mı? Ben de.
Bölümümüz ile Buenos Aires Üniversitesi Ziraat Fakültesi Hayvansal Üretim Bölümleri arasında yürüttüğümüz bilimsel işbirliği kapsamında hocamız bugün çalışma alanıyla ilgili bize bir sunum yapacak. Şu anda seminerimiz hem Zoom üzerinden yayınlanmakta hem de Buenos Aires Üniversitesi YouTube kanalından canlı olarak yayınlanmakta. Bu yüzden bundan sonraki konuşmalarını İngilizce yapacağım. Sunucumuz da sunumunu İngilizce yapacak. Sunum sonunda sorularınız olursa hocamıza sorabilirsiniz. Biz tercümanlık yapacağız. Şimdiden geldiğiniz için hepinize teşekkür ediyorum. Başarılı bir sunum geçmesini diliyorum. Well, dear guests and attendees, firstly, I would like to thank everyone for being here and participating our seminar within the scope of scientific collaboration between Dicle University Agriculture Faculty and Buenos Aires University Faculty of Agronomy Department of Animal Production. Before starting the seminar, let me to give information about our guests and attendees. Now, our ambassador is here. Our Vice of Rector, Professor Dr. Kadir Turan is here. Our Dean, Professor Dr. Miktat Şimşek, and also researchers from Faculty of Agriculture and Faculty of Veterinary. Some veterinaries, directors from Provincial Director of Agriculture, and employees and students. I would like to point out that our seminar will be broadcast live on Zoom and Buenos Aires University YouTube channel. Today, we have a guest speaker, Professor Dr. Daniel Felipe Salamone. He is full professor and director of biotechnology animal laboratory, UAB, Universidad Buenos Aires de Argentina. Also, he is principal researcher of the National Research and Technology Council. He is member of Academy of Agronomy and Veterinary Science of Argentina. Dear Professor Salamone, on behalf of my university and myself, thank you so much for coming here, being with us. He will present his seminar entitled Animal Cloning Technologies, Frontiers in Assisted Reproduction of Domestic and Wild Species. Now, I would like to give brief information about Professor Salamone. Professor Salamone got his bachelor's degree in veterinary medicine faculty in Buenos Aires University. He got his MSc degree at University of Saskatchewan, Canada. He received his PhD degree at the University of Massachusetts, USA. He has been in Japan in 1991 as a postgraduate researcher and 2015 as a researcher fellowship. He was the director of cooperation project with Vietnam and now Invest Bank. He was the director of Department of Animal Production, Agronomy College of the University of Buenos Aires. And he was president of the International Embryo Technology Society. Since 1993, he has given several courses about cloning and transgenesis, supported by the Argentine Brazilian Center of Biotechnology with travel hours and scholarship for selected Latin America students. Dr. Salamone has published more than 70 referred papers, has represented more than 100 conferences articles in more than 20 countries, different 20 countries. The main topics of his research are embryo micromanipulation, gene edition in bovine, horse, sheep, goat, cat. He is also interested in assisted reproduction in white mammals. Now he will start to his seminar. After our seminar, please don't hesitate to ask questions to Professor Salamone. Yes, Professor, welcome. Again, thank you very much for being here.
Thank you very much. Thank you to the authorities, to all the persons that are present. And I will try to do a presentation, not very big, not very difficult to understand, and so everybody can follow the topic. As you know, we have this cooperation project with Buenos Aires University. Buenos Aires University is big, it's 300,000 students, and with more than 10,000 foreign students. Our faculty is small, with small number, 1,000. They are all around Buenos Aires City. Buenos Aires City is I think, a very interesting place because it's a very cultural and it's a very European city in South America, but with a Latin American flavor. No? I don't know if you know tango and football, Messi, Maradona, all of them, they are our people now. The Argentinian, the Pope now is Argentinian. And our university, although we are in South America, that sometimes we have chronic economic problem, we produce in Buenos Aires University 4 of the 5 Argentinian Nobel Prize, 2 of the Argentinian Nobel Prize were peace and 3 were in science. and two were doing his work in Argentina, and the third one in Cambridge, although he went to Cambridge when he already had the degree. So it's very interesting for exchange. The other interesting point about Argentina, and mainly about our university, that Argentina is a great provider of food for the world, because we produce 10 times more food than we really need. So we have a lot of exportation in soy. And the good point in some of our chronic crises, in which we are in a really bad situation and it's like the end of the world, sometimes technology and agriculture say our destiny, as happened with the transgenic soy. We have a big crisis, and just 5 years after, we were very, very good situation, and now again we are in a big crisis. So bad. Well, my department is animal science department. You can turn out. My department have an important group in animal breeding. They were helping to improve some specific breed in Argentina. I don't know if you know, but Argentinian meat is very famous because it's very tender. And well, in the last 30 years, we have a genetic program for improve this breed using the last technology, even molecular technology, and today is very good. They also have several groups working in nutrition, but we are open to explore different animal production, silk production, or honey production. Argentina is the second exporter after China of honey. And also we have a group working in nutrition that Musafer is very close, and I hope that soon you are going to have the visit of our director of department that is working in aspect of about fish production and ecology in the water related to agriculture lands. So I would like to start with my presentation, just showing you our department is at the left. Our university this year has 200 years of life. Here you see our team department, and what is amazing about our university, that Buenos Aires is a 3 million person city surrounded by Great Buenos Aires with more than 50 million of the persons. Most of the people in Argentina, they are living around Buenos Aires, but our university is very green and look like you are in a farm. Well, first of all, we were talking about embryo. Everybody has the concept what is embryo, that you have the female and the male part, and most of the person maybe don't know that the embryo is floating in the first 7 days at very least in all the species inside of the utero or the ovidum, inside of the female genital tract. So that gives us the possibility to take the embryo, to take out from one animal after the conception, and to take from a good animal and to transfer one animal without value. So in this way, we can optimize the production of one animal of high genetic, or if you have some interest. So the first way to produce the embryos is producing in a live animal. So the protocol is just use media to wash the uterus to collect the embryo, and the embryo are microscopic. So you should search in a stereo microscope and take the embryo. You can wash the embryo. If you wash 10 times one embryo in a Petri dish, you can avoid most of the disease. That was my first subject study that you can export embryo from one country to other with, one embryo have few cells, is microscopic, and also is packaged with a capsule that the name is zona pellucida. So the embryo can be also frozen. For this reason, if you want to exchange genetic between countries, embryo is a perfect way to do it. You have 100% of the genetic, and the other advantage that usually you have local disease, and this local disease, if you move one live animal, sometimes it's dangerous because can pass away because they are not ready for the new environment. But if you have a recipient that receive an embryo, the embryo at the time of the delivery is going to receive the defense, the antibody from the mother, and it's going to adapt to the new environment. So we have some technology that can improve the use of the embryo. We can take small sample, small biopsy, using micromanipulator. I'm going to present a micromanipulator in a while. And with this sample, we can detect the sex, we can detect disease, the genetic value. So it's very interesting. And we realized right away, some years ago, we have done a work with horse. Horses are very important in Argentina. Our country was made with horse, no? Because it was the way to until 100 years ago. So, but the horse keep today in racing horse, jumping horse, and we have polo horse. Polo horse, as you know, polo is a game, needs some specific speed, some special ability, like Maradona, no need to move and change the direction and go faster. So the polo player prefers just female. As way to go to the future, we start to do embryo sexing, and we produce the first sexy born animals, because polo player like females. So, but today we know that we have this microchip with large number of testing. You can know in some way some disease, as I told you, you can know, and we hope that you can select by performance in the future. No? So this kind of simple technique to take a piece could be very useful. The second technique that I want to talk is one that I really interested since I learned several years ago. As you see, I was younger at that time. I visited Japan, and in Japan, I learned how to produce the embryos in the lab. So here the technology changed, because you should recover the oocyte, the gamete cell, the female gamete, and the oocyte can be recovered by ultrasound today. But at that time, we collect in the slaughterhouse. We used to go to the slaughterhouse, get the ovary, aspirate the oocyte, as source very cheaper to produce embryo in the lab. So when I start to do the number, how many embryo I produce in the lab, I realized that in a couple of months I produce 20,000 embryos. So that is very impressive, because the numbers are very large. And what you have when this in the top, you have the oocyte. In the second panel, you have the oocyte at the time of the fertilization, and after fertilization, you have just one cell. If you modify these cells, you are going to modify the future animal in all their cells. But you can keep in culture, so these cells divide in 2 and 4 and so on until produce what we call morula. and later a blastocyst that is already have blastocysts have two parts. One is going to produce the placenta, and the other is going to produce the fetus. As you see, this is the capsula. This is the membrane that surrounds the embryo and the oocyte, and is called zona pellucida, that protect the embryo and keep together the cell. And we know that we start to play with this factor. This is one lab that is our lab that is expensive, but it's not so expensive if we are going to start to talk about the technology that we can use this specific lab. You need, of course, an incubator to culture the oocyte. These are stereomicroscopes that you can see and manipulate the oocyte or embryo. And this is a micromanipulator that basically is an inverted microscope with this tool. When you move this joystick in the other side, and you move a lot this joystick, like in the other side, you move just a little. You transmit a microscopic movement. With this, you can take out the nucleus of one oocyte, you can inject one sperm, you can do several things. So let me tell you this kind of play that we started years ago, that producing in vitro embryos, how it can move quickly to the industry. Well, here you have the total worldwide embryo production, and here you have the in vivo, the first technology that I described you, embryo production, and here you have the in vitro production. One is when you collect a live embryo from a live animal. In the second, you collect the oocyte, you produce the fertilization in the laboratory, and you culture the oocyte until the transfer. Look how was increasing. What is interesting that this increase was in Brazil, because some of my friends started to do this technology that we were using in European breed in Cebu. And they realized that Cebu had twice of number of oocyte than the European cows. So this increase is more than 200,000 oocyte per year at the very beginning in a country from South America. Several years after, this increase was copied 10 years after by state. And today, in vitro production of embryo is relatively important. In Europe, still is the numbers are low, but you have several places that you have company that they are doing in vitro production of embryo. In Argentina, in the last 4 years, the in vitro production embryo go from to be lower than in vivo production to be higher. And today you have so many companies. The good point is to do the in vivo production, you need veterinary, you need a stereomicroscope, of course, a very good method, a very clean technique. But to do the in vitro production of embryo, you need a lab, you need a hood that filter the air. You need incubator, and generally you need the biologist or a person that is specialized in the manipulation, that understand. You need a team. So it's a good opportunity for companies. It's a good opportunity for start up. Well, as you see, these results are from the country that report every year the number of embryo producing in vitro or in vivo. As I have been part of the International Embryo Transfer Society in the last 25 years, as told you, Musafer, I was lucky to be part of the board, vice president and president, And we try to encourage everybody to report their results because we understand where go the industry, where we should invest, where we should do the research. Turkey, still we don't have report, and for sure you have practitioners that are doing well. If any person report to our committee, is like we don't have the result. What happened in this country? The second technique that I want to talk today is more complicated. In the technique, the in vitro production of embryo, in vitro fertilization, we put the sperm with the oocyte, we keep together for some hours, and we produce the embryo. In this technique, it's like we don't wait that the sperm go by themselves to the oocyte. We inject the sperm. So this technology was developed more than 25 years ago, and the experimental animal was the human. Gian Piero Palermo, an Italian doctor who was the first baby produced by ICSI. Today, you have more than 2 million persons that were delivered thanks to this protocol. So, if you have almost a country that person produced by ICSI, that means intracytoplasmatic sperm injection, we are going to talk about ICSI. I thought that at very least we should explore in animals. I was lucky to have several students. I didn't realize until was the 25th anniversary of the first baby ICSI baby, and Palermo invited me to do a review about this technology in domestic and wildlife animals, and I want to show you some of the results. But you know, it's interesting when you are working with different animal models, because we tried to do the ICSI in cow, and it was not working very well. We tried to produce sheep ICSI babies, a calf, lamb, and we were very ineffective. We produced one lamb from almost 70 embryos, so it's very unsuccessful. But we start to realize that each reproductive technology is very good in some specific technique. For example, in vitro fertilization is working in cows, but when I want to do in vitro fertilization in horse, it's impossible to get an embryo. No fertilization at all. We still don't know what is wrong with the horse oocyte, but we can produce an IVF embryo. We can produce, of course, animals. But when we start to do ICSI in horse, we have the surprise that they develop very well. And it's interesting because you remember the first kind of the technology to wash in vivo embryo and to get in vivo embryo. If you want to do in cows, you can treat the cows with hormone and you can collect several embryos. But if you want to do this in horse, it's impossible because the anatomy of the horse doesn't allow at all to get several embryos. So horse is a species that regular superstimulation and embryo collection and embryo transfer is not working, where regular collection of oocyte and in vitro fertilization is not working, but ICSI is working fantastic. And you can tell me what could mean for one university work with ICSI. Well, let me tell you what happened with Texas University, where was working one professor that is my friend, Catherine Hinrich. Catherine Hinrich started to do horse in a state, and they produce like 2 million years. They sell like 2 million ICSI in just a couple of year. They start, and for 2 years, every year they produce 2 million. And what advantage have to do the ICSI and the horse? That you can get several embryos in vivo, you can collect several oocytes. You cannot fertilize, but you can do ICSI. So the good point, and it's not the easy technique. It's like every micro manipulation technique is not like impossible, but it's like to play the piano. You need to practice because you are working with microscopic structure. The other nice point about the horse is that you can take oocyte and you can sell for 24 hours by mail, even without any temperature. You can receive your oocyte, you can keep one night on your decks, and then the other day you can mature this oocyte and 24 hours after do the in vitro fertilization. So it's very convenient. Now in Europe, you have in Italy just a place that is doing ICSI. But let me tell you how you start with a new technology. For example, I'm sure that here in Turkey, you have very good physicians or biologists working in human reproductive technology that perfectly can move to do ICSI in species like horse. No? And now some place like Qatar or Egyptian government, they really want to have this technology, and they don't have. Some of the advantages, if you have a very expensive straw, you can chop, because when you inject, when you are doing in vitro fertilization, you need maybe million. But when you are doing ICSI, you need just one sperm. So you can chop an important semen that sometimes is very expensive in horse and use it. Even if they are dead, the sperm, and they don't move at all, sometimes the DNA is okay and you can produce embryos. Well, we start to explore in different ways. We want to produce transgenic animals years ago with genetic modification. And we have a wonderful tool at that time that is a gene that is green fluorescent protein, that if it's expressed the gene, you have some color in the animal. You literally can produce an animal that if you illuminate, it's going to glow. No, we use at that time as tool to develop the transgenesis technique, because we were optimistic about some applications. Some of them we are going to show later, but we have a kind of evolution of this kind of technology. Well, at that time, I produced for a company using Clonign a group of transgenic animals. But the truth is like they should invest like $600,000 to produce this animal. So we were working in a relatively poor university in terms of the financial support. So we decided to decrease the value. And then we thought at that time that to use the ICSI combined with the introduction of gene maybe was a solution. So we made this paper that I was particularly proud, that we produced ICSI embryo in 5 different species, and we tried to inject a construction that with a strong promoter and with green fluorescent protein, and we produce beautiful green embryo in 5 different species. The problem is when we want to produce the deliveries, we work with the wrong species because we try to do mainly in cows and mainly in sheep, and sometimes you pay this mistake and this lack of knowledge maybe with 5 years of work. So we couldn't produce our transgenic animal by ICSI, but we are going to come back at this subject when we are going to talk about gene edition. So, as I will tell you later, we were working with cloning horse, and when Why? Because Argentina is one of the main exporters of horse meat, export mainly to Japan, to Holland, the meat of horse. So we have a source of ovary, and we start a cloning project. But when we got the first clone, and the people realized that the one of clone, not our clone, that was brought from a state, was sold in $600,000, the slaughterhouse told us, you want ovary to do cloning, and after they sell the animal this value, please spend $10,000 in ovary per month. And we say, we can do it. So they say, okay, let's go to do, we produce our clone at that time. I will show you the story later, but we decided to start to do the oocyte collection by ultrasound. No? You go in transvaginally, you can see the ovary, you grab the ovary, you can see the ovary, and with a needle, you can aspirate the follicle, and you collect the oocyte, and after you can do, for example, ICSI. Well, this is our first work, of course, that we try to collect information because I know that the public is heterogeneous. I don't want to go too much in detail, but it's easy to understand when you have a baby. You know? This is our first ICSI horse, you know, that was produced by some of my PhD students. Of course, sometimes we have dream and we say, it would be wonderful. We're the expert in cloning. Why we don't clone one sperm? We produce. And how we can do that? Of course, if you fertilize one oocyte, you have two nucleus. How about if I take the nucleus of the oocyte and I leave just the nucleus of the sperm? What happens is like I copy the nucleus of this sperm, and I can test genetically. How this the sex and all the genetic, and use just that sperm that can produce what I really want to do. No? So this was a dream, and we were doing this protocol with ICSI, and after we were doing with IVF. And we produced a first paper with ICSI. We have a second paper ready to be published. My dream was to have a delivery. We don't have. But maybe nobody is interested in this one. No? But I think that sometimes you can have a cell line, not just few copies, but a cell line you can characterize, and maybe after you are going to produce, it's a new way to sex the sperm. So this is the work just to explain you. This is the time we fertilize. Look, this is a fertilize. We take out just here is a zygote. We eliminate the female pronucleus. We allow the cell division, and we use this cell to produce a new embryo. The PhD student that just graduated 1 year ago, I want to produce a calf, but she was more enthusiastic about some basic aspect, and we finally didn't got the animal. Well, this is a micromanipulator again. No? This is a joystick, and you move this part, you have a small movement in the other part, and you use pipette. As you were looking at before that I have a pipette, this pipette is just to hold the embryo or the oocyte, depending what you are doing. This second pipette look is sharp. You can go through the zona pellucida. You can stain the nucleus. You can aspirate the nucleus. This is the basic of the cloning, to take out of the nucleus from one oocyte. This nucleus is replaced later for a nucleus of a cell, generally a cell from the skin. What is the advantage of the cell of the skin? That you can easily culture in a petri dish. It's just one man, you can have several million of these skin cells you can preserve, and if you produce animals from this cell, they are going to be from the same sex, and they are going to be same genetic, and so you can, if you really want, reproduce indefinitely one animal. Well, we transfer the donor cell in the enucleate oocyte. This here is the program to build all the animals. So what gives us the oocyte? The oocyte gives us a factor to reprogram. A cell in the skin is programmed to be a cell of the skin. But to be an embryo, they need to express different genes. The enucleate oocyte have all the factors to make that this cell reprogram and believe or behave as an embryo. Okay, to be in touch, one of the ways is to fuse by electric pulse. I don't want to go in detail, but it's like we are doing the cloning like in 5 different ways. We are doing the cloning, this is the way that was produced Dolly. The problem about this technology that I think, if you pick up the right cell, wonderful. If you pick the wrong cells, sometimes the animal don't have a thymus, or sometimes the animal are without the testis. Why? Because the gene that produce this organ were not right reprogrammed. So for this reason, we decided the other thing that you can do in place to do the fusion is you can inject the donor cells. A third way that we have to produce is to eliminate the zona pellucida, to enucleate the oocyte without the zona pellucida, which is much, much easier, and after attach to a donor cells with a drug, and after fuse. The problem that the zona pellucida keep all the cell together. If you are without the zona pellucida, the cell spread, you don't have a live embryo. So we need to rebuild the zona pellucida, and we are doing with a small hole in a plastic culture dish. So we put here one structure, and they are going to keep together. But we start to think, if we put one embryo, why we don't put 2 embryos together? Or why we don't put 3 embryos together? It's not the case. If one of the structure fails to produce one organ, maybe the other is going to compensate. And so this, in fact, was working really well. And the other nice point, from production point of view, we make a team. I have one student that is doing the nucleation with them, and another 2 that working with the stereo microscope who do the attachment and the fusion. Well, you need to know several aspects from cellular biology, and one of the aspects that when the sperm go in, induce that the embryo start to develop. And you don't have a sperm in this case. So you need to mimic the sperm, and you can do this one or with some physical way, like electric pulse, or you can do it with some specific drug. We specialize in some way in different drugs to induce this development. Here is the experiment, one of the experiments we have done. We put just one structure, just one enucleate oocyte with a donor cell, or 2 or 3. So we take out the zona pellucida, we eliminate the nucleus, and we transfer donor cells. And after we put together in this small hole. And what happened that all the structure integrate in just one. So we produce these wonderful embryos. We transfer this embryo, and this is our first core that were produced starting with 2 embryos, and this one starting with 3 embryos. And what we see that when we transfer a single structure, we have like 30%, 40% of pregnancy. When we transfer any delivery, when we transfer with the aggregation of 3 embryos, we have 70% of pregnancy, and we have deliveries. Well, this is what sometimes we scientists love to see, especially when you are in a Latin American country. It's like to see your horse or your experiment in the cover of a prestigious journal. No? And well, this is interesting because it was a Criollo horse. Our first clone horse was a local breed. Was brought for the Spanish, raised in the wild, and after was redomesticated. And it was the first Argentinian clone polo horse, and this was the first champion horse. Well, in Argentina, we produce a company that survived for some years, but a couple of my students produced the company Clonign, that is the top company in horse cloning in the world today. And this is an independently cloning company, but Argentina is famous because it has the more number of clone horses in the world. What is interesting is like help a lot to the polo business, because the polo open from Argentina is very prestigious. And there was 2 teams, one made by clones and the other for no clone, no? Even with their own fan. No? The fan from one team that they were all different, they say, we are different. Of course, we are going to win. But the truth is like win the clone team.
Yeah, Daniel, sorry, time is.
Well, so we are going to go quickly. We have been doing cloning with cheetah, tiger, zebra. We can talk later if you have questions. And we are doing cloning with this technology of embryo aggregation of yak. This is, I was telling the open that a person play the game changing with the clone horse. And finally, I want to talk a little bit about chimeric embryos that we started to produce several years ago, to try to have several copies. We split one embryo in several cells, and we produce tetraploid cells. Tetraploid cell can be done by fusion of 2 cells, and when we aggregate, they go to produce the placenta. So we have now several projects that we try. For example, if you transfer a buffalo in a cow, with buffalo embryo in a cow, they are not going to survive because you have immunological rejection. But we have the capability to produce the cell of the placenta with this tetraploid embryo, and in the way that when we aggregate, that is similar technology that we were using for the cloning, the placenta is going to be from one species and the fetus is going to be from a different one. And very quickly, for a very long time, we tried to produce, and we produced transgenic cow expressing in milk human growth hormone. The idea was to use a bioreactor to use this animal, this recombinant protein, to produce medicine for dwarf persons. With the production that we have admitted, we calculate, this was our first project in combination with the biotech company. We tried to produce by using transposon technology. It's a different way to produce transgenic animal, also using one enzyme and helping the insertion of the transgene. We produce one lamb that were transgenic for anti-hemophilic protein 9, died very soon, but our idea was to use as bioreactor the animals. But we had very, very low success rate until appeared a new kind of technology that is the gene editing technology. Gene editing technology, you use an enzyme and there with a RNA guide that allow to recognize gene and cut as a seizure. With this technology, we eliminate the gene in embryos for PRNP gene, that is the gene responsible for prions, for mad cow disease. And the truth is, like, we not just that, we can insert in the specific locus a new gene. And that was the first work that was done doing cut and paste a new gene in a specific place. And finally, we are working now trying to produce, to edit pig to generate organs. We have several genes that we should switch or change, or some of them eliminate or insert, to reduce the rejection of pig organs when they are transferred to humans. And we are thinking about heart and kidney, and we are thinking about the pig as donors of these organs. So this, we are now in the step that we have the embryo. We didn't have the gynecologist, and right now we have the full capability. We are going for our third transfers in pig. And the thing that we try again was the ICSI, but now for gene addiction, and we were successful 100%. Well, finally, to the end, with all this technology, we have our kind of zoo of animal, no? Our first IVF embryo that we produced back in '93, our first sex set, our first ICSI, and this ICSI lamb, and we helped to do the first clone dog. And well, these are our transgenic cows and clone and transgenic cow for human growth hormone. And we think that all this technology can help, and we have opportunity. This is a project that I helped years ago in Brazil to save a swamp deer that we were moving to a place for protection. And in this case that you capture animal, you have opportunity to take sample, and this technology that I described you can be very successful. No? This, we have a different team of PhD, and now we have several seniors in our group. This is our group right now. And we have previous students because we started to work like 22 years ago. No? So thank you for your attention. I'm sorry that.
Thank you very much for your nice presentation, and we are happy for being you here. Muchas gracias por venir aquí. Estamos mucho gustados por aquí, te vemos por aquí. Çok teşekkür ediyoruz Sayın Hocamıza. Genel olarak Profesör Daniel'in konuşması bir dakika için özetlemek istiyorum. Buenos Aires Üniversitesi'nin yaklaşık 350 bin öğrencisi olduğunu söyledi. 20 binden fazla öğretim üyesi, araştırmacı bulunmakta. 200 yıllık bir üniversite, şimdiye kadar 5 tane Nobel ödülü almış bir üniversite. Çalışmalarını özetleyecek olursak hayvancılıkta biyoteknoloji çalıştığını görüyoruz. Embriyo transferinden tutun da klonlamaya kadar gitmişler. Hem canlı üzerinde hem de in vitro olarak çalıştıklarını görüyoruz. Yine endüstrinin ihtiyaç duyduğu konularda yoğunlaştıklarını görüyoruz. At klonlama, embriyo transferleri yaptıklarını görüyoruz. At eti ihraç eden bir ülke olduğu için özellikle atlar üzerinde çok çalıştıklarını görüyoruz. Yine son projelerinde de insanlar için organ üretebilecek, domuzlarda organ üretebilecek projelerin yürüttüğünü görmüş olduk sunumunda. Çok teşekkür ederiz dinlediğiniz için. Sorularınız varsa sorularınızı alabiliriz şu an. I just summarized your presentation to our audience. If anyone has a question.
Uzaklığı konusunda ne aşamada öğrenebiliriz. Programların şartları üzerinde çalışmalar var. Akciğer ve de etkisine kadar
Ask to your last project that on pigs produce donors. What is your on which stage?
In the stage, we have the capability to produce the different embryos, and we are transferring this embryo. We're producing this embryo in different ways. We're producing in vivo fertilized pig. After fertilization, and we collect the zygote and we inject at one cell stage. We are successful in the development of blastocysts. We transfer blastocysts, and we are doing very well with ICSI. We inject the sperm with the CRISPR system, and it's working fantastic, like 100%. And we produce the blastocysts. So we transfer 2 animals with this 7-day blastocyst, we didn't get pregnancy in these 2 animals. So we switch now, we are doing the transfer with just 1 day of culture, less than 1 day almost culture, and we transfer to the oviduct by surgery, and we made the last surgery last Friday, and this Friday we have again. So, so the idea is to have soon the edited animal, because if the embryo is edited, but anyway, it's like we were expert in embryo in vitro production. We didn't have a gynecologist, so we should prepare our person, and we were like that 3 months ago with 2 transfers, and we sent to one of my students to Murcia University. They are the top notch in embryo transfer. And so we transfer, we do successful, I hope, but this is the stage. But I didn't talk with you, but why it's important to help in one university with this kind of session. They are between applied and basic science. Well, from my lab, we have like 5 startups that were done, that of course they are selling product. For example, one is Kairon, is the company, we don't have a relation with Kairon, they were just our student, but they just sell clone horse. We have another company that is Hijo, the name, that means son in Spanish. Hijo is producing in vitro fertilized bovine embryos. So, and they are doing with more than 20,000 calves per year. We have a third company that is a company that started with gene edition tool, and they used to detect They worked for a while in our lab. Was one of my PhD students that he didn't have place to continue with his research, and he brought his team to my lab. And an American company bought the company and paid to my institution $4 million for the company, and we received 500 with 500 dollars. So, but for us it was bad. But the point is, for the country is good, no, because they have like a place also in San Francisco and in Buenos Aires. They are doing research, and they're using genetic edition for COVID detection, and so they are doing well. And the company that is in relation to your question, the company is, we have a company now in our lab that is new organ. No? And so we receive, the quantity for this project is not so big. We receive like 200,000, not me, the people from my lab, $200,000 to do this pig, and well, we should work hard to get the second round of investment, no, because to have the animal, everything is much more.
Thank you. Buenos Aires.
So, Professor, it's a privilege to host you here. As a layman, of course, I'm not able to follow up all the seminar, but I'm more into the economics of this in vitro reproduction process. As I understand, you are, this business, this scientific research, scientific work is shaping up the today's and feature of the animal reproduction. So a rough estimate of yours, the global business portfolio of this in vivo, in vitro reproduction, what's the size of this business globally, and which are the leading countries in this in vivo, in vitro reproduction?
Okay.
Just translate to the students.
Sayın Büyükelçimiz sunumu dinlediğini, özellikle de bazı konulara dikkatli çektiğini söyledi. Hayvancılıkta in vivo, in vitro çalışmanın ekonomik düzeyi hakkında hocaya sordu. Global düzeyde gelecekteki etkileri ekonomik olarak neler olabilir, bu konu hakkında ne düşünüyor diye sordu.
Well, in some way, I think that in society you have different roles. I don't know if it's roles. Different roles. You have people who do the business, and sometimes people that just dream, and at the very end you finish poor. And I was dreaming like with in vitro production like 30 years ago, and now it's business. So, but for me it's like worry. And with the ICSI, it was, and the cloning and everything is the same. Let me tell you, the one leading country now was Brazil. And why was Brazil? Because they started to produce. The business is not so big. It's $some million for the companies. It's $some million for the breeders. No? But it's good for the country, because what you are doing mejoramiento, you are doing improvement, genetic improvement. And genetic improvement means that you are going to produce more milk,
more productivity,
more productive animal, because you are working, in general, you are not working with the regular breeding, you are working with the breeders. We have a specific name for who the business is not to sell the meat or to sell the milk, is to sell this one, but to sell the genetic, to use the top animals to bring animal from outside. So I think I can do this calculation, but I think that the impact is big. But the other thing that I want to take advantage of your question is like we have all new brand new technology. For example, this gene edition. For example, with gene edition,
Please, short.
okay, with gene edition, this pig, if we can sell this pig for a herd, you have people with an allergy to pig meat that maybe can be consumed. So you produce like a hypoallergic pig, and you can do this one with the milk to produce a hypoallergic milk without some allergens. And so, and I don't know, but you have genes that you have animal with more muscle. So, and what I think that is more important, what is the value to avoid a disease in a society? Tell me today that is maybe different we thought before. So we can make animal resistant to disease with just a genetic manipulation. So I think that, and if you have the in vitro production, I'm dreaming in that today. Again, probably we will be a business like in 10 years, and then we'll lose this business.
Her şeyde oynuyor.
Evet, her sorunun cevabı bir seminer kadar olduğu için.
Evet. Yani iki konu da tam net açıldı aslında.
Konuyu tekrar açıklayacak olursak bu konunun ileriki 10 yıl içinde de sadece hayvansal üretimde artışa değil, aynı zamanda özellikle bir takım hastalıkların tedavisinde de bir çare olacağını düşündüğünü ifade etti. Ekonomik düzeyde de in vitro çalışmaların biraz daha ekonomik olacağını ve bu yöndeki çalışmaların da daha yoğunlaşacağını hocamız ifade etti. Biz bu sunumu tabii yapacağız, kayda aldık. Bunun Türkçesini de hazırlayacağız. Özellikle öğrencilerimize, katılımcılarımıza gerek web sayfamızda da gerekse başka şekilde ulaştırmaya çalışacağız. Yes.
Dr. Guram from Faculty of Veterinary Medicine. I'm very happy to see you here, and thank you for your excellent presentation. Actually, my main area is food science, but I know I see some of my colleagues from Department of Artificial Insemination, and I would like to ask you about water buffalo. Have you had any experience? Because I know that some of my colleagues have really difficulties when they are having their projects of water buffalo. So can you please let us know if you have some experience? Thank you.
Sure. Yes. Well, butter buffalo, we just start to work. We are lucky to have a breeder in the Northwest that give us the semen and all the support that we want. And what we try just now is to be familiar with in vitro production of hybrid cow, because we don't have a slaughter cow close to us, so we are collecting bovine oocyte, and we are doing the fertilization with buffalo. So we are producing hybrid embryo. And the other thing I didn't show you because I didn't have time, the other thing that we are doing is like we start to produce tetaploid cow embryos and we put together with the hybrid. And so we have integration. Well, now we are in the step that we have first table with the hybrid embryo production comparing to more specific, second table with the complementation, what is going on, what's happening is amazing. How we improve the blastocyst production with the complementation. And next step is to transfer. And first we're going to try to produce hybrid cow. I don't know if it's useful or no. Maybe it's wrong. But it's easy for us to do that. And after people are doing in vitro production of buffalo. But we realized that we have a lot of problems. We have problem with the sperm, for example. We have a very bad sperm and relatively good sperm. Even we are thinking to use now like unfrozen semen. But it's true, it's like our knowledge is limited, and you have just a couple of groups in my country using, and you don't have so many breeding. But we go in this direction because we think that like a special production, not to produce mozzarella is good, no? To have this.
Thank you. Şahan hocamız veteriner fakültesinde manda tohumlamada, suni tohumlamada arkadaşların çalışmalar yürüttüğünü söylediler. Onunla ilgili bir takım zorluklar yaşadıklarını ifade etti. Hocamız da kendilerinin böyle bir proje yürüttüklerini, özellikle hibrit elde etmede, embriyo elde etmede, transferde bir takım teknikleri başarıyla yaptıklarını ifade etti. Teşekkür ediyoruz. Buyurun Sayın.
Yes, Professor Salamone, I would like to thank behalf of my university and the University of So I have also so many questions because it's very interesting. And I appreciate your work. So my question is the embryo transfer fertilizer rate, because you mentioned about the horses and it's about 60% of the fertilizer rate of success. I would like to learn what about the cow. For example, in Turkey, it should be about the 50 or 60%. France knows better than me. So I would like to quiz about what's the rate of the success in the cow. Thank you very much.
With in vitro producing.
Yeah.
Well, first, it could be fresh or frozen. If it's fresh, generally good companies is more than 60%. We have 60% in our lab, 60%. If you are transferred fresh in vivo, maybe 70%. If it's frozen, it could be between 30 and 50% of pregnancy. Depend on the company, depend on the technology. Yes, but sometimes you can play, because in hot place, usually it's worse the development during the hot season. But if you are doing it in the lab, sometimes you can compensate. The natural is low, and so because you are doing in vitro, the oocytes are keep and the embryo in vitro are not very hot, and you transfer the blastocyst. So sometimes you can do better than with natural breeding. Anyway, all these technologies for good genetic.
Okay, thank you. We have time after our seminar upstairs. We can go on. The last question.
Thank you very much. I'm not any scientist actually. I will ask you a different question related to social. If this technology is widely increasing in next, so it's very complex technology, especially Clonign, right? So how is it affected environmentally, and how is that affected environmentally of animal life and including the animal sex life?
Well, the truth is like it's interesting. It's look like now nobody is really. Most of the country, they are not interested in cloning. Cow cloning, less interest. Why? Because you still have a problem during parturition. Generally, you need cesarean C-sections. You have high mortality, and it's very inefficient. Anyway, I would like to try the technology that we're using in horse. I would like to try in cows, in the efficiency of it. And also, we have done some experiment using undifferentiated cells. So efficiency could improve, but in the short term, I don't see people interested to clone cows. Just some interest in horse and not too much. Because when you have a clone, you have a twin. You don't have a better animal. But if you select two good animals, you have an improvement, genetic improvement. So I'm a kind of not very optimistic about the future. For example, now I'm involved with some dog cloning company. Why? Because people want to have don't care. They want to have a similar pet. So in terms of reproduction, wolves were cloned and were without testes. So bad for reproduction. And also some banteng were cloned and also with reproductive problems. So it's not cloning, it looks like it's worse for reproduction. And so I think anyway, I have when I produce in wildlife animal, for example, we produce a zebra or we produce a tiger embryo. So it's like we have a tiger embryo deficit. And so we are using as model because we want to control gene expression. And we know that the clones are wrong, but we try to generate now a new technology to arrange the gene expression. And so we are doing this basic research. But the truth is, like, I would like to try the technology that we were using in horse and look what happened in terms of all the parameters. Because all is with the dolly technique, not with this new technique.
Thank you very much again. Hocamızın semineri burada bitti. Kendisine tekrar teşekkür ediyoruz. Kapanış konuşmalarını yapmak üzere öncelikle Sayın Dekanımız Profesör Doktor Mihtaç Şimşek Kürsü'ye davet ediyorum.
For the attention, for the time, for the question. Beautiful question. Thank you. And it was
Dean Vice Rector, Professor Dr. Kadir Turan, Distinguished Professor Dr. Daniel Salamone, Distinguished Deans and Vice Deans, academic institutions, managers and participants. Welcome to the Agriculture Faculty, Dicle University. Dear participants, we are pleased to host Professor Dr. Salamone at Dicle University Faculty in Engineers in Agriculture. Dr. Daniel Salamone worked as professor at the Department of Animal Reproduction at the Faculty of Agriculture at the University Buenos Aires, Argentina. Professor Dr. Daniel Salamone has a lot of research and animal clone technologies. He's one of the most important scientists in his field, the world. He gave a very important seminar on animal cloning technologies, printers in assisted with reproduction on domestic and wild species. In the presenters about the participants. Thanks to Professor Salamone for presentation this seminar and coming to our agriculture faculty, Dicle University, Turkey. Dear participants, I would like to give some information about Dicle University Faculty of Agriculture. Our faculty was with 1992. Agriculture faculty has 7 departments. Undergraduate students, horticulture, plant production, crafts, animal science, and agriculture nations and technology engineering. No student, farm structures and irrigation and agriculture economics. Dear participants, the number of undergraduate